The overall goal of this procedure is to compare the tissue-specific migratory abilities of 2 or more cell subsets in the same mouse recipient, allowing the analysis of many tissues and cells in one experiment. Here, we show a method for comparing the migratory abilities of in vitro generated gut-tropic and control T cells in a wild-type recipient mouse. The method can be used, for example, to determine the ability of gut-tropic T cells to migrate to the intestine, mesenteric lymph nodes, peripheral lymph nodes, and spleen compared to control T cells. First, gut-tropic and control effector T cells must be generated in vitro. Next, the experimental and control T cells must be labeled with differential dyes and mixed in a 1:1 ratio. Then the cell mixture, except for a small aliquot used for confirming the input ratio, is injected into the recipient mouse. Finally, the mouse is sacrificed 18 hours after the adoptive transfer, and the organs of interest are analyzed for the presence of gut-tropic and control T cells by flow cytometry.
The main advantage of this technique over existing methods, like adoptive transfer of single-cell population, is that a combination of different subtypes is injected in the same recipient mice, greatly reducing the variability associated with intravenous injection or from using different recipient mice. The implication of this technique extends toward therapy of diseases associated with massive leukocyte infiltration, such as inflammatory bowel disease. For instance, it allows us to determine homing molecules required during normal as well as during aberrant lymphocyte migration to the intestine. Furthermore, this procedure allows us to assess the efficacy of drugs designed to block leukocyte trafficking.